So we are moving on to the next session, where it's all about application of all the data that has been generated within Bovrec, mainly work package 2 and 4, into genomic prediction models. And I think I can speak for the whole group of work package 7. It was really a nice interaction between work package 2 and 4, so it's a nice way of working together across disciplines. And so for all the quantitative geneticists involved, so I can really say, so we've really learned a lot. And I would like to introduce the first speaker. Emre is the work package leader of work package 7, and he has, together with Lucians, developed the software to incorporate the genomic features for genomic prediction, and will talk about their study in Denmark.
Thank you for the introduction. So far we have heard about what type of data was generated in Bovrec and how it was generated. And there has been some questions about, okay, how do we use this in breeding, or what do we tell the breeders to exploit this information? And then I will not give you a complete answer, but at least we will find some information that could be useful to move forward after the Bovrec project. Is this one? So I will start with giving a bit of context, and then data processing and methods, and some results from Danish breeds, Jersey and Nordic red, and some conclusions, and maybe how we will keep investigating on this issue. So it's a part of, as Birgit said, it's a part of work package 7, where we actually try to combine the information that was generated in the previous work packages. Yeah? Okay, sorry. Yeah, so we will. So we try to combine the information that was generated in the early work packages, and how we could actually use this information in practical breeding schemes. So we know actually that it's been investigated a lot if we could make use of whole genome sequence data in genomic predictions. Initial attempts actually were to use them in genomic predictions as they are, but what we have observed is actually you gain really nothing by just using all this whole genome sequence information in genomic predictions, but there should be some smarter ways actually to include them. So what is being done currently is actually to add some more SNPs, as you heard in the morning, to the standard 50K SNP chips. What is basically done is that you have 50K SNP chip as an additional, as a random component, and then you have these SNPs that are selected from these omics analysis as another component in your genomic evaluations. And sometimes these efforts lead to actually customized SNP chips where you have also these additional SNPs in your official evaluations. But adding these additional SNPs into genomic evaluations, maybe it's easier for EQTL or from the QTL analysis or MetaGWAS, but it's not straightforward how you actually include these ataxic pigs, because it's a very wide region. And I have talked to Marcy from Alberta, and she said that there is like 30 million SNPs that needs to be included on top of the 50K SNPs. Of course, you have to reduce it down to make it manageable, but there is not a procedure that how you should actually do or how you select these SNPs. So I decided not to follow this common approach where you actually add some SNPs on top of the 50K SNPs, but do something different, like enriching the 50K SNPs with the results from omics studies. I will tell a bit about the procedure that we followed, but I wanted to stick to the 50K SNP chip because it's already in use. And if we stick to 50K SNP chip, then it will not require a lot of changes in the pipelines of routine or official evaluations in the cattle breeding. So it can be implemented in routine as a way to develop, as straightforward as just weighting SNPs in standard GBLUP or single-step GBLUP analysis, and Arash will talk about single-step analysis later. So what is needed is actually just to run additional analysis to obtain SNP variances, to use them as weights in your genomic analysis. So we studied with the Jersey and Nordic red dairy cattle and with the mastitis. I got the Delegates proofs derived by Luca, by Arash, and then I used them as phenotypes, and I had about 9,000 Jersey, or 10,000 Jersey, and 30,000 red dairy cattle. And then I used 2018 sort of as a cutoff date, so animals born before that date, before 1st of January 2018 were in the reference, and the rest were in validation population. And we worked again with the 50K SNP chip. So what we did is that, I'm not sure if we have a pointer here, but it seems like, yeah, is it? Yeah, it works. But maybe you cannot see it. Okay. Yeah, okay, you cannot see it. Good, because it's green. So what we did is that we took the bottom line is the 50K SNPs. Okay, so bottom line is the 50K SNPs. What we did is that we went up and downstream, 1000 kb, and then we said, okay, if within this region for this 50K SNP, if there was a significant association reported from the early work packages. If this was true, what I call sort of like SNP information matrix, if this was true, we'd put a 1 for that specific entry for this SNP. If it was not reported as a significant SNP, or it was not in the pick or for the ATAC-seq analysis, then I put the 0 for that. So we have zeros and ones in this analysis, or in this converting step for the information from other omics data to the genomic analysis. So this is a diagram of how many SNPs were included in eQTL, MQTL, ATAC-seq, and from the QTL analysis, but there were so many actually SNPs that were included, or that were, I cannot say annotated, but yeah, they were assigned to be associated as an eQTL marker. So to exploit this information, so we have used this information matrix. We use this to model the variance per SNP. So SNPs in the same category, like eQTL, QTL, MQTL, ATAC-seq, whatever, then they will have the same variance. And this can actually be immediately seen as a way to use weighted GBLUP in your analysis, because in a weighted GBLUP, it's been theoretically proven that you can actually use SNP variances to weight your SNPs, and then you can immediately get SNP variances estimated from this model, and then you can use them in genomic BLUP or BLUP variations to weight your SNPs, so that you will have a biologically informed genomic predictions in the evaluations. So we tried this approach within the Jersey breed and the RDC in the Danish population. The RDC is the Nordic population. So we can see here that, these pluses means that on top of 50K SNP chip, I use the information that is from the QTL detection analysis or from EQTL analysis, MQTL, and so forth. So, and then at the end, I combined all of them together. And what we can see from here is that for Jersey, it worked pretty well. Each additional information actually improved the accuracies, and collectively, they improved the accuracies as high as 14%. But that was not the case for red dairy cattle. But I'm still looking into it because these populations have a bit of a different background. For example, RDC is sort of like a composite breed where you have three different breeds actually that are jointly evaluated for like 20 to 25 years. So there are chunks that comes from different breeds in their genome if you look into it. And also there's, it is composed of Swedish red, Finnish Ayrshire, Danish red, but there are some traces of like 10-14% of Holstein and some brown Swiss and so on. So that it's a bit of a mixed breed. So we got, as you just saw, we got some promising results, but this approach improved the accuracy for Jersey, but we couldn't really improve it for RDC. But again, there are different structures or different backgrounds of these populations, so I need to look a bit more into it, how I can actually get some improvement also for RDC. I think it could be related to the size of the size of the region that we selected for a QTL, like 1,000 kb. That was really arbitrary. And if you think of mixed breeds, actually this 1,000 kb may be even too large, that actually you don't have the LD with where actually the causal marker is or causal variant is. So maybe it should be narrowed down for RDC. I don't know. I'll try. But I will also look more into the pipelines of the whole RDC analysis to see if I can get an improvement for it. Because in November when I presented, actually, there was also no improvement for Jersey. Then I looked into it a little bit more, and then we got this accuracy that's improved quite a lot. Of course, the method we used, actually, it's not been really tested with data integration in this form. And the algorithm we implemented, it has also not been tested with any real data before. So there's a lot of things actually I need to, go and check and maybe need to fine tune and so on. But overall, I think it's been, promising to get these results. Okay, so thank you all for the efforts.
Thank you very much. Questions?
Jose Espinosa from the CRE. So I don't have any clue about the mathematical modelization, but will it be possible to, instead of just include this matrix of binary values, so just ones and zeros, to actually incorporate the strength of the signal, for instance, of the attack peak or whatever?
Yes.
And do you think this will improve the results?
Yes, it may improve the results, yes, but for some of them. But then we need to think of if we talk about like p-values that are really p-values in statistical sense, or if it's something different. But they can be added as well, yes. The model is flexible, so that's the sort of usefulness of this model that you can add any kind of covariate. It could be categorical or continuous that you can just add into the model to model the variance. So yeah, that could be possible as well. Thanks.
Renzo?
Hi, Emre, thank you. I would like to ask if you could reason around this, that this approach is quite interesting because it keeps the genotype of the animal close to the individual, because you avoid basically the imputation step, right?
Yes.
You only include this matrix of information. On the other end, what happens if you have a variant that has been identified, which is not in your, say, 50K? Do you rely on the fact that it's on LD with the other 50K variant SNPs? Or, yeah, do we just lose that info? So maybe there is a trade-off there?
Yeah, that's, yeah, there could be, yeah. So we may be missing some yeah, SNPs, whole genome sequence SNPs that, yeah, it should be tested, I think. But I don't know how many of them we are missing, actually. We have to check that. But we, again, I mean, so for the cows, we have low density chips. For the bulls, we have 50K. So there is an imputation, but from low density to 50K. But we are not imputing the whole genome sequence. And another advantage is that you don't really need to think about how you actually extend your chips each time you discover something. You know, it does not need to be included in a next generation customized SNP chip, which may not come in a short time or something. So all you need here is just to try to relate your finding with the 50K SNP chip. But that's a good point. I mean, we may be missing some, but I don't know how to verify this for ATAC-seq, basically. Because the others I may try to add, you know, additional SNPs, but I don't want to. When it comes to ATAC-seq, I don't want to add like 10 million SNPs on top of 50K and try to estimate with the same data 50,000 effects and 1,050,000 effects. Makes no sense.
Okay.
Someone in the audience raised her hands. Marcia, do you want to make a question?
Yes. Thank you, Emre, for your presentation. Do you hear me well?
Yes.
Oh, okay. So my question is about the bias results. You didn't show any bias results. I wonder if for the base LV it decreased compared to 50K. I mean, when you add other features.
I think bias and inflation.
No, I have not looked into it yet. I mean, I will look into it if, you know, it improves first the accuracies, then I will look for the bias. But if it doesn't improve accuracies, it's probably not very useful, and I don't need to go more in detail. But I will, yeah, I will look into it when I
Okay,
write
thank
the paper.
you.
Yeah.
Okay, then I think we move on. Thank you very much,
Thank you.
Emre.
Yeah.